Coming to you from Earth, this is the Virulent Vortex, a podcast in which we discuss infectious diseases from pathogens and molecules to populations, pandemics, maybe even space. My name is Jaap de Rode. I'm a biology professor here at Emory University, and today I'm joined by
Sethi, and I'm a fifth-year student at Emory University and in East Lowen's lab.
Well, welcome to the show. Thanks so much for talking to us about your research, which I believe is about reoviruses. Can you tell us what those are?
First of all, thank you so much for this great opportunity. So I work on this virus called, as you mentioned, reovirus, and the full name is mammalian orthoreovirus. To me, it's a very fascinating virus because it's mostly non-pathogenic, and all of us have at least once been infected with this virus as kids, and we'll probably even keep getting infected with this virus. So it's a very it's universal. Everywhere you find this virus.
So you're saying that I probably have this virus? Yeah. And so what does it do?
In very few cases, it causes severe disease, like gastric issues. So as kids, when we had cough and cold or a tummy issue and the doctor said, you have a viral disease, it's probably real. And the reason I think it's perfect is because it replicates and survives without harming its host. So it's kind of made it. It's reached the perfect optimum.
So it's a little bit like flu virus, except that it causes less disease and therefore it's more successful.
Yeah. It is a respiratory and enteric virus.
Good. And so what specific thing do you study about these viruses?
So I study this phenomenon of collective dissemination, where basically viruses disperse in groups rather than as single virions, which deviates from the general norm of one virus per cell.
So what you're saying is that normally when you think about viruses going from cell to cell, there may just be one viral particle that makes connection with a cell and then goes inside. But you're saying these viruses do it together, like a group.
Yeah, almost all viruses do disperse in groups in one way or the other, whether it be in the form of, they're inside these membranous vesicles, or they cluster onto bacterial cell surfaces, kind of hitching a ride on those bacteria to kind of reach its destination of infecting the host cell. It also aggregates in the environment. Reo was actually found to even aggregate in water. And that's just Reo. We don't know. There's so many other viruses there. HIV aggregates, influenza aggregates, VSV aggregates in saliva. So it's a very common phenomenon, which I think really needs to be studied and how it impacts infection.
And so what is in it for the virus to go together? Do they need each other?
I guess all of us need each other. Viruses do. And there have been actually several studies showing how virus aggregation can be very beneficial for the virus in terms of stability. There have been experiments where they have clumped the virus and then exposed them to disinfectants and radiation, and they've seen the virus remain infectious for a longer period of time. In fact, the polio vaccine, that was the one where they inactivated the virus with using formalin, and it caused polio disease in the participants. And there was a paper in the 70s that came out and said that it was they actually kind of did the work to show that it was polio aggregation that prevented complete inactivation of the virus by formalin.
Interesting. So there's protective, yeah.
That's one of the benefits.
Okay.
And of course, from my research, we've seen that it leads to more viruses entering the cell, larger burst size, and an interesting sneak peek to the data, although my paper's yet to come out, but it actually produces a really diverse progeny. It doesn't need to infect a lot of cells, but it is yet able to produce a diverse progeny, which is an important requisite for evolution.
Yeah.
So for the virus to evolve.
So because they're in groups, they're more likely to exchange genes with each other. Is that the idea?
Yes.
Yeah.
Genetic material with each other.
Yeah. And so how do you study this? Do you do this in Petri dishes, or do you infect cells or humans? What do you do with this virus?
So my system is currently in vitro. So the mode or the type of aggregation we're doing is we are manipulating the pH and ionic composition of the solution that the virus is in. So the idea is that every viral particle has a charge, And once the charge is neutralized, it is able to overcome the repulsive forces and come together. And we've seen through transmission electron microscopy, we've seen these viral particles aggregate when we manipulate the pH. We've also seen these viral aggregates entering the cell as aggregates.
So that's cool, but I think there is something that you find even cooler, and that is microorganisms in space. So tell us about that.
Yeah, that's hopefully one day I'll get to work on that. So what I want to study is how viral transmission and disease and evolution might pan out in a microgravity environment. In fact, I am currently writing a review article that will discuss these points, and also how collective dissemination might actually help the virus to survive better and evolve in microgravity environments. Because, as much as viruses are great as for a researcher, like for me, the way I see it, but it can also be super dangerous because they can cause disease. Yeah. So as much as the astronauts are at risk, there's also potential hazard of bringing those variants back to Earth.
Yeah, and I was going to ask you about it. So you think about viruses in space, that is within man-made environments, right?
Yes.
So these are spaceships and space stations. Yeah. That's okay.
It's like we carry viruses, plants carry viruses that are also at the space station. Animals have gone to space. They carry viruses within them. And there have been actually a lot of papers published that have shown astronauts shedding dormant viruses. So basically, there's a lot of activation of the dormant viruses like herpes, cytomegalovirus, varicella zoster. So when they're up in space, these dormant viruses activate, and they shed a lot of this virus, which again is super dangerous.
Yeah, and so that's dangerous to the other astronauts, and but also you said, so when they come back to Earth, so is it that in space they may recombine or make new variants, and then when they come back to Earth, that would be a risk?
Probably, but again, we don't know the evolution rate, the mutation rate of viruses in space, and I think we really need to study that because viruses can be really dangerous.
Okay, so, and to study these viruses in space, how are you going to do that? You're going to go up there and study them?
If I get a chance, yes. But, I think the new idea is of the new commercial space station is kind of have a digital twin system. Like there'll be equipment systems at the space station, and a replica of that will be on the Earth. So we wouldn't probably have to go there to do the experiments. They're trying to automate a lot of that, because being in space is also, it's not a great thing for the humans too. Our bodies take a lot of toll. If you want to do long duration space missions, it is very risky. But yeah, if I get a chance, why not?
Yeah. So if someone says, do you want to go for a few weeks, you'd say,
oh,
sure.
Yes, 100%.
I'll come and hunt for some viruses.
Oh, sure. Yes.
All right. Well, that sounds like a really fun experiment to do.
Yes.
So, well, thank you so much for sharing these exciting ideas with us.
Thank you.